Hi. Hi, everybody. Sorry for all the delay, but we got started, and that's great. So just please make sure to put your phones on silent before we start. And as you've heard, this meeting is being recorded, and we usually upload the recording on YouTube a couple of days after. So that's the recording disclaimer. And this is the schedule. We're running about 10 to 15 minutes late, but hope that you don't have very tight commitments after. So we're going to start with some announcements, especially about May events. And we're going to have a trainee talk and then our main speaker, who most of you have met already. So this is just our organizing committee. We have a lot of students on the organizing committee, including some faculty, and these are the faculty up here. So these are our upcoming events for the rest of the season. So we have on March, Dr. Karen, and on April, Dr. Yongjin. And for May, we have a new event. It's going to be a late-term competition. So we're excited for this, and it's going to be at Langara College. The top line summary of it is that it's going to be open to all undergrad and graduate students from all institutions, whether SFU, UBC, Langara, or somewhere else. It's going to be a presentation, something bioinformatics related from computer science side or biology sides. Anything is good as long as it has touches on one of these fields. And there's going to be a 3-minute hard limit. And the purpose of this is that to try to communicate your research to the non-expert audience, the least expert audience that you can imagine. So no jargon, just try to distill your idea. We're going to release more details about the competition sometime, I think hopefully by end of March, maybe early April. With registration, we're going to announce it on vanbug.org and also in the email list. Just as a reminder, we're at the end of the season, which means we have a new season coming up with new speakers. So if you have suggestions for speakers that you would like to see, go to the website, suggest a speaker. We always take these into consideration when selecting new speaker slate for the next season. So with that, we have to thank our sponsors, of course, the graduate program in bioinformatics at the UBC and SFU, Genome BC, Connexia, Langara College, Stem Cell Technologies, and St. Paul for allowing us to be here. And as always, VanBug is affiliated with ISCB and 2 other bugs, Montreal and Toronto's. So with that, I'm gonna just quickly introduce our speakers. So second in order, we have our featured speaker by Dr. Hosna Jabbari. So Dr. Jabbari is an assistant professor in computer science department at University of Victoria. Before that, she was an assistant professor at University of Vermont. And before that, she was between Vancouver and Victoria, got her PhD at UBC Computer Science Department, and her alma mater is UVic, right? And Dr. Jabbari's interests are in RNA, but also more focus on finding diagnostics and therapeutics through RNA research, and she leads the COBRA lab, what was the COBRA lab stands for? Computational Biology and Research Analytics. So I'm going through this quickly because we're short on time, but we're going to go to our student speaker, Carolyn. So please have it. I'll just... you want presenter mode, right? Yes. Yes. Here. Okay. Just getting use the mouse if you want to.
Here, those are fine, so that's gonna work.
Okay, so I just stand on here and I think they should work. Okay. So take it from here.
Hi, everyone. My name is Carolyn Risley. I go by Kara. I'm a PhD candidate in Stephen Jones' lab as part of the Genome Sciences Center, as well as University of British Columbia. The talk I'm going to be giving today is fact-checking cancer knowledge bases using natural language processing. Okay, I'm going to start with why we want to use NLP for fact-checking. Fact-checking data manually is extremely time-consuming. Journalists verifying a single fact can take anywhere from a couple of hours to even several days. And as humans, we make mistakes, both when we're entering data and when we're reviewing it. So this has become an area of significant interest in the NLP community, mainly due to the spread of misinformation. And this can actually, there's been some studies saying that like stuff on Twitter that's fake can actually spread faster than the truth. This means that fact-checking things promptly is very important. This is also useful in fact-checking data that is produced by other models. So for example, information extraction models, which extract data from the web, we need to make sure that that data is correct as well. And finally, most topically, there is a well-known problem called hallucinations in large language models, such as ChatGPT, which I'm sure a good portion of the audience has tried at this point, where it produces things that sound coherent, but they're not actually factually correct. I've given it or shown you an example here from GPT-3, showing that it created this story and timeline where a woman gave birth after she died. Now, both of those sentences make sense on their own, but when you put them together, well, that's just not possible. And a human can see that pretty easily. A computer has a lot harder of a time. And a lot of the research so far has focused on making these things fluent and sound correct, but hasn't got all the way to make sure they're factually correct. So that's one of the areas that's coming up right now. So how can we use this in cancer research? I'm going to start by introducing you to the Personalized Oncogenomics program, which is part of BC Cancer. POG was a landmark precision oncology trial, which involved whole genome, whole transcriptome sequencing of metastatic tumors. Similar to other precision oncology trials, we begin with obtaining the patient consent, followed by whole genome, whole transcriptome sequencing of their tumor biopsy. We then, once this is sequenced, we then need to produce a report that is presented to the molecular tumor board. Now, a molecular tumor board is a group of people with various expertise and backgrounds. It'll have pathologists, it'll have clinicians, it'll have bioinformaticians, and we all work together for this. Following the tumor board, any actionable targets, which were identified, are then used to potentially inform treatment of the patient. Now, what we are most interested in today is going from the sequencing to the actual report. Well, much of the process to create the report is automatable. Some of my previous work has worked on that, such as calling variants or running various bioinformatics pipelines, the process of the actual variant interpretation and what we take away from people's variants. This is mainly manual, and it represents one of the largest bottlenecks in this whole process. Variants need to be annotated, interpreted, and summarized using literature review by the case analyst in order to create the report. And there are a number of things we want to know about a variant. Clinically, the most important tends to be the therapeutic implications. Will this variant change the treatment options? Is the variant linked to sensitivity or resistance to any of the treatments we were previously considering? But we also want to know things like the biological implications of, does this variant change the function of the gene, and will it have any prognostic or diagnostic implications for this patient? So why do we need to fact check these knowledge bases? So cancer knowledge bases have been largely successful at reducing the interpretation bottleneck that I just described. They're used to automate this process of annotating variants with the therapeutic, biological, prognostic, and diagnostic implications. Because we're using these in an automated form, it's really important that they have a high degree of accuracy. We can't have those hallucinations that you see in some of the in some of those other models? Well, it might be funny when they're producing a story. When it comes to medical information, it's significantly less entertaining. So generally in knowledge bases, like for example, Civic, which I'll be talking about, content is submitted, but then it still needs to be reviewed by an expert before it is accepted. So this means that we can crowdsource to enter things faster because there's a low level of expertise required, but then we're still creating a bottleneck when we're trying to make sure it's correct. And as you can see on this right-hand plot, this is things that are being entered, submitted, accepted, and rejected, and the submitted number is quickly outgrowing the number of items that are accepted. And finally, there's a dearth of scientific data sets in the fields of fact-checking and natural language interpretation. So we are just kind of uniquely poised to be able to contribute that with access to expert personnel. So most automated fact-checking systems consist of 3 parts. The first is claim detection. So this is where if you have an input text, you need to determine what is fact-checkable, like what is opinions or what is actually things that we can actually verify. The next step is that you need to retrieve the evidence. Now, for our purposes of scientific fact-checking, the corpus we often refer to here is going to be something like PubMed. So first we need to determine what articles are relevant, and then from those articles, we need to determine what sentences or excerpts are going to be either supporting or negating the claim we're making. And then finally, the final step is once you have that evidence and you have the claim, they're input into some sort of model which does the claim verification. This really gives it the label of whether we think it's true, it's false, or we just can't tell. To explain how we can use general fact-checking model to check cancer facts in a structured database, the first thing I need to go over is how natural language inference is used for that claim verification. So this isn't the only way you can do the claim verification, but it is probably the most widely used, and it's the simplest conceptually. So what happens is you take the claim text and the evidence text, you concatenate them into one long string, separate them with some sort of delimiter, or as in here, you can see a token. This gets put through an encoder model so that it has a numeric representation that models can understand. Gets put into a second AI model, which does the classification. And here you can see that I've put a really simple false claim for you where the cat is blue and then it's not a natural color for a cat. So this should have given it a label of refutes. And although there are a few scientific and expert curated data sets, there are several in the more general domain. So here I've listed quite a few of the more popular fact-checking data sets. And where ours really differs is that a lot of these go from unstructured text to the evidence being in structured like a table or unstructured like a paragraph. Whereas ours will be taking, because we're fact-checking a database itself, we'll be putting in structured text and then fact-checking it against both unstructured and structured, which is slightly more challenging in some ways. So this is an example of taking an entry in the Civic database. This is from the Civic database. This entry states that this variant KRAS G12D mutation is associated with sensitivity to sotorasib and RMC 4550. Below, I'm demonstrating how we linearize that into a text representation that we can then put into the model. The corresponding evidence for that claim would be excerpts, as I've shown here, which are from the paper where the authors mention these facts. Now, these can also be in a table. The ones here I'm showing are just text selections. The last bit of background I'm going to go over here is that in order to introduce you to the experiments I've performed, I first need to go over a little background about transfer learning. Now, transfer learning is the idea that you can train a model on one task and then reuse the model to retrain it for a new task. You might ask, why would we want to do this? Why not just train the models directly? Similar to how humans learn, an AI model might benefit from learning English more generally before trying to predict, for example, if a YouTube comment is inflammatory. But really, with enough data, we don't need to do this at all, which brings us to the crux of the issue. labeled data sets are incredibly hard to create. They require the expertise we've described, and it's just really manually time consuming. So by using transfer learning, we can leverage these vast amount of text from these large unlabeled data sets and then use a small amount of curated data. This was demonstrated most notably in 2018 with the BERT paper. In our experiments, we are using the general language models that are first trained on these large corpuses like the PubMed or Wikipedia. And I've shown you which models we're using here. And then we are fine-tuning those models on these natural language interpretation data sets, which contain these pairs, the claim and the evidence. Finally, in a second stage of fine-tuning, we're training it on the data that we have collected, which we have called Civic Fact, because it goes with the Civic database. The first results I'm going to show you evaluate how well our model works before we've trained it on our own data. How well can we get this to work just using these publicly available data sets? So what we've done here is I've taken those general language models and I fine-tuned them on the publicly available data, and then I've evaluated what data we've collected so far to see how well it performs. Now, since there's 3 labels, random would be about 30%. So you can see some of these are worse than random, which is not great, but to be honest, a bit expected. We don't expect this to perform very well without a data set that is actually tailored to what we're planning to do, which we just kind of wanted to verify what that is. Here, for each of these, I have taken either doing it with all of the data sets at the same time or excluding one and only using 3 of the data sets. Now, the reason we've done this leave one out approach is so that I can tell which data sets are the most helpful for what we're doing. As you can see above, the Feverus data set has the biggest impact by quite a large margin. That's not really surprising. This is the data set that I showed you in the previous one where it had both tabular and free text data, which is most similar to what we're working on. And it is also the one that is more of a fact-checking data set. But it does indicate that that's the one that we should keep if we're going to be using it in our fine-tuning. What was a little surprising, however, this data set here, actually the model improves when we remove it, which indicates that it might be contributing some negative transfer in that what the model learned before really isn't helpful. And that is surprising because this, out of those 4 data sets, is the only one that is based, is only on the same domain. It's the only one that has science related data in it. We hypothesized that's because it was partially automatically generated. And it was actually a set of multiple choice questions, which was using heuristics reformatted into this type of data. So it probably contains some unnatural text formations. The final analysis I'm going to talk about is the multi-stage results. So these are those NLI models from the last slide, but we have then further fine-tuned them with the data set we've collected so far. This is really promising because this data set is pretty small. So the fact that we're able to get this high of accuracy is pretty nice. You can see, right, I don't know if I can get to that one. Under the PubMed BERT model there, we're getting 87% accuracy in predicting the labels for the claims. This is on the initial data set, which was smaller, and we're actually collecting a lot more data still now. I'm running out of time, so I'm not going to go into too much details. There are a few limitations to this. We have a 3-class label, and one of the classes is significantly smaller than the others because it's less useful when we're trying to collect that data. When people want to collect data that either refutes or supports a claim, that's really useful for inputting things into the database. They can collect that and use it as they go. If they just want to have the 3rd class that is not enough information, doesn't tend to help them out as much, so that data is being collected slower. I would like to verify these labels with a 2nd round of annotation. We're planning to next train the evidence retrieval model. Currently, we're only working on the claim verification, and then we'll be publicly releasing this data set. No research is done in isolation, and I am particularly fortunate to work with a large group of people, both at the University of Washington through these civic curators and editors, as well as through my own university and at the Cancer Center. I'm funded by CIHR and UBC, and that's it.
Any questions? I do have a question. Do you think that like there is a certain accuracy level that people start getting comfortable with for something like this field?
I think when it applies to medicine, we're probably never going to be fully comfortable with using AI for most things. What I envision something like this fact checking doing is that it'll take away all the really easy decisions. So we'd use it to make sure that people aren't making stupid mistakes or things that are like completely the wrong paper they input and they said, oh, this variant is related to sensitivity to some drug that isn't even mentioned in the paper. Like those should be pretty easy things to catch. Whereas the really tough ones where it's a bit subjective about, oh, do we think this is really convincing evidence or is it just like maybe they should have done another experiment? I think that those are going to be passed off to the human reviewers still. And as they are done, the model will improve and maybe eventually we'll trust it enough to use it for most of it.
Do you kind of weight your evidences? Like, for example, from one journal versus another journal, they might be doing different facts. So
Yeah.
their evidence might not be like well defined as the other journals.
Yeah, so that's something that the Civic database has included in it. So they have two tiering systems. They have one talking about like how big the study is, if it's preclinical evidence or if it's like a clinical trial that was done, they're going to rate those differently. But they also have a slightly more subjective field you can enter, which is like a rating on the evidence, saying like, were they reproducible experiments? Like how many experiments did they do? Were there things that they should have covered that they didn't? And that goes from like 1 to 5. Usually you end up with like, it's pretty easy to pick out a 1, it's pretty easy to pick out a 5, and everything else kind of feels like a 3, and 2 and 4 are kind of hard to, grade. But yeah, that data is included as well. And that's probably something we'd be looking at later. Right now, we're just trying to make sure that the facts that get entered are actually something that the authors claimed in the paper.
Can you please go back to slide?
Which slide, sorry?
12.
12. Yeah, I totally know how to do that on here. Oh, I went too far. No, that's it. Okay.
The evaluation data set is the Civic data set, and the models are basically like trained on each of them, excluding one.
The models are trained on different. They're not trained on the Civic data set.
Yeah.
They're trained on these public data sets. Yeah, that's correct. What was your question?
Yeah.
Oh, okay. Any other questions?
Any questions over from Zoom?
Great.
I maybe have a concluding question. Do you can this like model also spit out like how confident you are in those evaluations?
Yeah, so there's some measure of confidence that you're able to get out of the model as well. Right now, I mean, that would be based on the number of times you've made that prediction, and well, this data set's kind of too small for us to do a lot of that confidence intervals yet. But yes, there are ways of doing that. I haven't looked into it in too much detail yet, though.
Cool. Okay. Well, thank you so much. Okay. So if you have any questions throughout our speaker's talk, I think your preference is to have them as they go instead of waiting to the end and like you missed something in between and like causes you to like not understand the rest of the rest of the talk. So same for the Zoom. If you have questions, we have Zoom moderator and she will let us know if you have any questions. So with that,
thank you so much.
You're welcome. You can use the mouse or controls.
Perfect. So I will be talking about unraveling RNA function through structure prediction. And the first slide goes over something that all of us have seen at some point in our biology class. The central dogma says that, okay, DNA holds our hereditary information, and this is used to make proteins, and proteins are the molecules that do all the activities in our cell. In this view, RNA is merely a transfer for information to create proteins. But recently, we have seen that this RNA is not just the transfer or write for information. It has many active roles in the cell, and a lot more that are being discovered with the new molecules. So it's interesting to find out what does this molecule do and how does this molecule work. So let's start from the beginning. We all know the double helix on the right side of the DNA information that holds all our heredity information. RNA is very similar to DNA, but it's a single-stranded molecule that is copied from the DNA. It has two distinct ends. We refer to them by 5' or 3' end. And this RNA is just like a sticky tape. Because it's single-stranded, it folds back to itself, forms a lot of complex shapes. And these complex shapes are the ones that predict their structures or make the structure, the function of RNA. RNA structure is therefore important when we want to understand its function, or we want to design new molecules for different work in biotechnology or for vaccine development, for example. RNA structure can be as complex as the ones that you can see over here. This is the structure of RNA in 3D. If we flatten the structure, you can see the two-dimensional structure or the most familiar planar structure of RNA. And as you can see in this structure, this sequence is comprised of 4 nucleotides. These are adenine or A, cytosine represented by C, guanine G or uracil. And these bases form hydrogen bonds together and pair. And these base pairs are the basis for RNA structure. When we talk about the structure, we talk about different levels of information. When people refer to primary structure, they merely mean the sequence of RNA. So a string of ACGUs. And this information can be stored in a string or a vector, for example. When we talk about secondary structure, in addition to the sequence, you have more information. These information are about which base pairs with which pair. And when you talk about the tertiary structure or 3D structure of the RNA, we add more information. And this information is about geometric information of the structure. Ultimately, what we want are experimental methods. They are the most accurate and give us the best view of RNA's structure. However, these methods are time-consuming, they are expensive, and in some cases, they are not even possible. So the structure of RNA changes when, for example, they put it for crystallography. That's why computational methods are indispensable for RNA research, and majority of computational methods focus on secondary structure prediction. Secondary structure prediction is much easier than tertiary structure prediction, and but it sheds light on the 3D structure of RNA. When we talk about RNA secondary structure, we can further classify these structures into 2 main groups. One is pseudo-knot-free structure, or the ones on the top, and the other ones are the pseudo-knotted structures. Pseudo-knotted structures are more complex, and their prediction, of course, is going to be much more difficult. The one that you see on the bottom of the slide, I don't know if you can see, no? Do you see my, no, okay. The one on the bottom of the slide is a famous structure that is referred to as kissing hairpins. Okay, thank you. So this is this structure is a kissing hairpin structure. As you can see, there is a hairpin over here, and there's a hairpin over there in 3-dimensional space. These two come close together by these 2 green hydrogen bonds, and this is an amazingly hard structure to predict. So when we want to look or analyze the structures, we can look at the structure in different views. This is the most familiar structure of an RNA in planar graph. This RNA has two distinct ends. In here, you have the UNA over here. If I take the two distinct ends and pull apart the RNA sequence, so that sequence forms a line, but the hydrogen bonds are still intact, I'm going to get a structure like this. This is called an arc diagram. The backbone is shown from 5' end base number 1 to the 3' end base number 55 or 54, and the hydrogen bonds are shown with arc. I can show the same structure in dot bracket format. This is another format that you see most often when you are analyzing a structure. In the dot bracket format, unpaired bases are shown with a dot. The left bracket, the open bracket, shows the 5' end of a base pair, and the closed bracket shows the 3' end of a base pair. So opening is the open bracket, and closing of a base pair is with a closed bracket. When I show that mathematically how the structure is, how a pseudoknot-free structure is shown, is that we say there's a structure R, and in this structure, IJ are two base pairs. So for every two base pairs, IJ or I prime, J prime, they are either disjoint, so they're not at all close to each other, or they are nested in here. So I prime, J prime here is nested within IJ. If I do the same thing with a pseudo-knotted structure, then you will get a structure like this one. So exactly the same thing, hydrogen bonds are intact. And as you can see over here, the hallmark of a pseudo-knotted structure is crossing base pairs. So the two long-range interaction over here in green are shown with crossing base pairs over here. Because we have crossing base pairs, if I want to show this structure in dot bracket format, I can't use it with one type of bracket. I have to introduce different types of bracket depending on how complex the patterns of pseudoknots are. In this case, two types of brackets are going to be enough. As you can see, these all are in normal bracket, and this is a square bracket that I'm using for the crossing base pairs. Or if I want to show the structure mathematically, I can say that there is at least a base pair in this structure R in which two base pairs are going to have crossing interactions over here. So if a structure only has one crossing base pair, then this structure is going to be considered a pseudo-knotted structure. The problem for secondary structure prediction then becomes, given an RNA sequence, you want to predict the secondary structure. So you only have sequence information, and based on the sequence information, you want to find the secondary structure. There are multiple approaches for doing that. Each approach has a basis for their prediction. The one that we focus over here is an energy-based, because with an energy-based method, you can predict the structure of RNAs that you have no information for. So unknown families of structures can also be predicted. But of course, every model has its own assumptions and restriction. In the energy-based model, we assume that RNA forms a structure that has the lowest free energy. Justification is it's more stable, and that's the structure that's going to stay. But we also assume that this structure formation is in isolation, so with minimal or no interaction with environment. And we assume that the energy of the total structure is the sum of the energy of its parts. So energy is additive. The challenge here becomes, how can we pick the minimum free energy structure among the exponentially many possible structures that can form? So over here, I have a planar graph for an RNA structure that has most of the features or loops. Hairpin loop over here, here, and here has one emanating stem. Internal loop has two. Multi-loop has more than three. And there are two special types of internal loop. That's a stack and bulge. And the energy of this structure is then sum of the energy of all the hairpin loops plus energy of all internal loops, multi-loops, bulges, and stacks. How tractable is this problem? We know that if we allow for pseudoknotted base pair, then predicting the minimum free energy structure is going to be NP hard and not even approximable. So bad result first. But there are some polynomial time algorithms that
Code red, Providence Alpha Bravo, level 10. Code red, Providence Alpha Bravo, level 10.
yeah?
Code red, Providence Alpha Bravo, level 10.
We okay? Okay, it seems to be okay. Okay, so there are, it's going to be a memorable talk now. There are, let's not get to conclude. There are polynomial times algorithms that restrict themselves to a specific class of structures, and that's why they can bypass this NP hardness or intractability result. For example, if we restrict ourselves to only pseudoknot free structures, we come to the very famous M-fold algorithm of Zucker and Steigler that has runtime of n cube and a space of n square. If we want to go for allow for the biggest class of pseudoknotted structures, then we get to Peanuts program of Rivas and Eddy. That's a 1999 paper, and this is the most general predictor for pseudoknotted structure, but of course is restrictive in terms of time and space. We have many algorithms that try to overcome this high time and space complexity. For example, our H-fold and iterative H-fold are based on hierarchical folding. They match the time and space complexity of M-fold, and the CCJ and Naughty are the ones that handle almost the same structures as in Peanuts, not completely, but they have lower time complexity. So let me give you a little taste of how this structure prediction in minimum free structure prediction works. So if we restrict ourselves to pseudoknot free structures and we want to predict the structure between point I and point J, so index I and J, we have only two possible cases. Either base I pairs with base J, in which case the energy of a structure is the energy of base pair IJ plus the energy of whatever structure we have from I plus 1 to J minus 1. Or I and J don't pair together, which means there is a point between IJ, we call it R, in which you can cut the structure into two independent structures. And the energy of the structure between I and J is going to be the sum of the energy between I, R, and R plus 1 to J. If we allow for pseudoknotted base pairs, in addition to the previous two cases, there might be the pseudoknotted case possible. So between I and J, I must pair with something, say L here, that crosses whatever J is pairing with. Roughly the time complexity of an algorithm like this is forced by how many points, border points, you have to keep track of to find this structure. If you notice with the two first cases, you have 2 or 3 borders. So the maximum is 3 borders, the time complexity becomes n cube. When you allow for even a simple pseudoknot presented in case 3, you have 4 ends of the border, and that's why the time complexity increases. So I think that's enough of algorithms. Let's see how we can use this structure prediction to find something fun and new about a famous RNA, for example. What is the most famous RNA these days? SARS-CoV-2. Hasn't it disrupted our lives enough for the past 3 years? SARS-CoV-2 is the RNA responsible for COVID-19, and it's a positive sense single-stranded RNA, of course, and it's considered a short sequence. It's about 30,000 bases. And while there are a lot of elements of this RNA are already known, for example, we know the coding elements and their places of SARS-CoV-2, And we know about some structures in the 5' and 3' end of the structure. There are still a lot of unknown parts for this sequence. If you consider viruses, or in this case, SARS-CoV-2 as a traveler, it has a limited space for things that it can carry with itself. So the 30,000 bases is not long enough. It has to make it compact in a way. How does it make it compact? It makes sure that it gets rid of all the redundant information, non-necessary information, and only pack the stuff that are going to be functional, important, and even use some tricks to make sure that it can reuse the same sequence to produce different types of proteins. So one of my students, so the first part of the talk is the work by Alison Ziesel. She's my PhD student, and this is the work that she did for her master's. She started by, she aimed to find out what are these regions, or can we identify some of these regions that have functional importance in SARS-CoV-2? How did we try to go for it? Was that she created a pipeline. The pipeline is shown on the right. And she started with collecting relevant data, similar data to SARS-CoV-2. Then she aligned this multiple sequences, generated the phylogenetic tree, and went through a couple of computational tools. Multi-Z, for example, takes the multiple sequence alignment and tries to find a region that might be functionally important. RNA-Z weeds out the low confidence regions and gives you the regions that are highly confident to have the structures. Then she added flanking sequence upstream and downstream of the areas that were functionally important, and reran the same thing to see whether there are structural similarities between these sequences, to assume that they were conserved through evolution of the virus. And she did that with three different tools: RNA-Z again, Locarno, which does simultaneous alignment and structure prediction, and Cacofold. Cacofold uses covariation, which is changing in the base pairs to keep the structure the same. So sequence changes, but the structure stays the same. And Cacofold allows for pseudoknotted structures. So the viruses that Alison picked are these 12 in addition to the SARS-CoV-2. As you can see, many of them are human viruses. Some are from bats, mice, and bovine as well. Based on her analysis, we found 40 regions with high confidence in function. Some of these regions are confirmed by previous publications, and some of them show conserved stems, for example. Some of the more interesting ones to me, at least this one, is a conserved loop that must be doing something that is conserved by evolution. And the one that I'm going to talk about more in here is this one, frame shifting element. Frame shifting pseudoknot is a key feature in replication of coronaviruses. What does it do? SARS-CoV-2 has two open reading frames. Normally, the ribosome only translate ORF1A, but sometimes there is a pseudoknot in the -1 PRF site, or a structure there that moves the ribosome one place back, so shifts gear, and then makes a fusion of these two ORFs, ORF1A and ORF1B, because these two are out of frame relevant to one another, and they can't be transcribed at the same time or translated at the same time. With this structure over here, there is going to be a fusion that is key for the replication of the virus. What do we know about this structure that facilitates this frame shifting? We know that this structure is this area that is considered to be frame shifting element is very much conserved within SARS, the original SARS, and SARS-CoV-2, the one for COVID-19. In fact, there's only one base difference between the sequence of these two different viruses. There's one other interesting fact, is that SARS-CoV-2 and MERS, the virus responsible for Mediterranean or Middle Eastern virus, have 100% overlap with their -1 PRF. But they don't share much of a sequence similarity. In fact, if you just check with the blasts with the SARS sequence, MERS is not even going to come up. And the other thing we know is that based on research on the original SARS, plasticity of the pseudoknot is going to be important for replication. What is plasticity? It means how many similar structures in terms of energy you have within vicinity of the minimum free energy structure, or how many structures are going to lead to the minimum free energy structure. So the second PhD student of mine, Luke Trinity, started with the two goals to work on the frame shifting pseudoknot. The first one was that because there is no known structure for MERS, and there was not a lot of information about frame shifting pseudoknot. Are we at code blue now?
Code red, all clear. Code red,
Okay.
all clear. Code
Okay.
red, all clear.
Okay, so Luke tried to find a structural similarity specifically between SARS-CoV-2 and MERS, and also set to explore the energy or structural landscape around the structures for the two to see what can we say about the plasticity of this pseudoknot. How did he work? He obtained the RNA sequence for both of them, calculated the energetically favorable stems, ranked the stems based on their free energy, and only picked the ones that were stable, meaning their energy was negative, and then used a program that we have called iterative H-fold that gets some information and some structural information and produces the minimum free energy structure based on that information, and produced secondary structure prediction for these viral sequences. So as you can see over here, we have these stems in dot bracket format ranked by their energy. So we refer to them as a stem 1, 2, all the way to 18 in this case. And these are the stems that ended up going to this secondary structure. So you can see multiple stems. So here, 1, 3, 9, and 18 that go with the lowest free energy structure. This lowest free energy structure, that is our prediction with this computational method H-fold, matches the known pseudoknotted structure, the native structure for SARS-CoV-2. And then we have many other structures that are energetically similar to this one. And we're going to explore them a little bit more to find out some interesting facts about SARS-CoV-2. So this, for example, is the structure predicted by iterative H-fold when the red stems were given as, these are stem 2, were given as input to iterative H-fold. So iterative H-fold was asked to predict the structure if we knew this stem formed. And these are the blue arcs that iterative H-fold at. These ones, the black arcs, are another structure that is energetically very close to the blue one, right? So if the blue one has a negative 15.2 energy, the black addition of the black one will end up with a negative 14.86 kilocalorie per mole, for example. Right? This small difference in the structure, you can't say which one is better because no energy model is perfect. So we have to look at what else we can find with these ones and which one of these structures are going to be important. For MERS, we did exactly the same thing. The only problem here is that we don't know anything about the native structure. There was no known native structure. However, our prediction matched the previously described structure for MERS. And we have the famous H-type pseudoknots that we saw before, as we did for SARS-CoV-2. The fun part about difference between MERS and SARS-CoV-2 was that whereas all the structure presented in the previous slide were pseudoknotted, in MERS, 6 out of 15 of the suboptimal structures have the pseudoknot, and the rest were pseudoknot-free structures. In general, the structures predicted were for MERS had higher energy compared to the ones that we predicted for SARS-CoV-2, perhaps indicating that these structures are less stable. And one other thing was that the suboptimal structures for MERS were not as close to the minimum free energy as the SARS-CoV-2 ones were. Perhaps this one shows that plasticity for MERS is not as much as SARS-CoV-2. Maybe that's going to indicate something about the transferability of SARS-CoV-2, for example. Then we start to look at, okay, now we know about the structures, and can we say anything about the mutations that can happen for this sequence? There were mutations on almost every base of this. We picked 7 of these cases. These 7 cases were either previously considered in SARS, original SARS, or we had some information about them being effective or ineffective. So some of them are positive result as changing the function or reducing the translation efficiency, or they were previously thought to do some kind of work with SARS. Out of these mutations that we have shown with red arrows over here, 4 are near the junction of stems. These are the U, C, U here, and these 2. And 1 is adjacent to an adenine bulge. That is the 62 one. And 2 are in loop situations. Some of these ones were shown to reduce frame shifting efficiency of SARS, as I mentioned. And we looked through to see whether the change of these bases can change a structure, which may mean that it changes the function. Oh, the way that some of these mutations are represented over here, so C13U is the C at position 13 was mutated to U, and similar to that. And these are only restricted to the sequence of frame shifting, which is 68 bases. So that's why you see these numbers from 1 to 68. So when we looked at mutated stems, so remember the pipeline, we went through the pipeline again, now with the mutation that we see these 7 mutations. Over here, you have the mutated base, and it's highlighted over here, and you see the different types of stems here. These stems, some of these stems removed some of the original stems or didn't end up with creating some of the original stems in the native sequence, and some other ones added some new stems that we didn't pick based on the original sequence. So over here, for example, in this U20C, you see there were 2 new stems that were detected over here. These stems were ranked based on the stems predicted by the native sequence, and they are nicely ranked in 5th position. So we have 5A and 5B. Some of these ones are higher energy stems, so they are in the bottom of the list, for example. We tried to do the same thing with MERS, and we obtained 237 sequences from NCBI. We only detected 1 mutation within the frame shifting region in 3 sequences, and we looked at the structures that were predicted based on these mutations. And in case of MERS, these structures remain the same. So in the previous one, these 3 structures, so these 3 mutations that are highlighted in red, didn't end up with the main native pseudonautid structure for SARS-CoV-2. And then we said, okay, there are some experimental data available. Can we use this experimental data to infer some more information about that? The data was SHAPE, so that's a chemical that you put on the RNA sequence, and the score that you get out of SHAPE is that whether this base is going to be paired or unpaired. It doesn't tell you any information about which base is going to be paired with. So we started with 4 different data sets. 3 of them are in vivo, and they are genome-wide, and the 4th one is in vitro, and it's focused on the frame shifting element. We developed a method called Shapeify. This Shapeify receives shape information as well as the original set of stems that we detected before, and tries to incorporate this shape data as a bonus to the thermodynamic model that we have for minimum free energy prediction to predict the structure. So we're not basing our prediction only on shape, because shape has noise. We're going to keep that information as add-on, as a bonus to our structure prediction. When we looked at structures predicted with shape, so Shapeify prediction, so over here you see the original stems, the stem 1, stem 9, and so on and so forth. They all ended up with so this is the native structure. If the 4 data sets agree on the coming from S1, stem 1 to the native structure, you see that with a black, and the color of this link between the original stem to the output becomes lighter and lighter if there are fewer data sets that agree on this transition. So as you can see with the native structure, we have multiple stems that can lead to the native structure. And in this case, we have some native adjacent structures, so they are energetically close to the native structure, but they have minor changes to the native structure. So in this case, for example, here we have in bold 2 base pairs that were added, and here in bold stars, we have 2 bases that used to be paired in the original or native structure, but now are going to be unpaired. So these are the minor changes that can happen to the structure, but the structure is going to remain within very close in the energy landscape to the native structure. Then we noticed that we can predict a lot of native non-adjacent structures. What are native non-adjacent structures? They are structures that can be predicted by some of the stems, for example, some of the original stems, but these ones are have a wider gap between these structures that were predicted and the native structure. So one of the fun things, if you compare the 2 slides together, you will see that these are clusters of a structure with these ones, whereas these ones, the structures predicted were very much tangled together. So much more ways or pathways to get to one structure in a native adjacent structure to the non-native adjacent structures. So there are more pathways to get to a native structure or a native adjacent structure than it is to these clusters of native non-adjacent structures. And in these cases, normally the consensus between the shape data sets was low. As you can see, not in every case we have a black line, for example. And the fun part here is that we're still looking for, over in this talk, I just went through one of these functional sites for SARS-CoV-2. Remember, we identified 40 based on similarity of them. And we follow the structure of prediction to kind of see or get some information about what can be differentiating SARS and the way it acted when you compare it with SARS-CoV-2, when you compare it with SARS or with MERS itself. And based on what we have found is that any changes to initial stem 1 for SARS-CoV-2 and MERS-CoV can result in changes in a structure, which means that the stem 1 is the most conserved one. It's probably the most functional site. And if you change that, you're probably going to see a change in function or reduced translation efficiency. We also showed computationally that there are multiple paths from the initial stems to the native structure, which can indicate something about plasticity. This plasticity was much higher in terms of SARS-CoV-2, not as high in terms of MERS. And we also showed that SARS-CoV-2 has structures of that are way more stable than the MERS one. Maybe that's the reason it can survive more. We looked at mutations. By looking at the mutations, we identified some of them that didn't reach the pseudonaut of the structure. For example, these two, because they destabilized the stem number 1, they didn't reach the native structure. But the other mutations, these other 5 ones, we expected that function is going to be the same because they didn't end up with a significant change of the structure in the mutated sequence. That while there were structural differences, the structural differences were not significant then. And redundancy in native and native adjacent path showed that there are multiple paths from initial stems to final structure, and also confirmed by shape was that if we disturb a stem number 1, because there were a lot of paths and a lot of significance going from stem number 1 to the final structure, we can have loss of function. So coming back to the central dogma, these are the Nobel Prize that were awarded to different discovery of different functions in RNA. And as you can see, many of them were awarded over the past couple of decades. It has been shown that computational methods can accelerate or help with identification of function of RNA. So maybe in the near future, we can expect some computational Nobel Prizes for identification of that. And I hope this talk has been inspirational for you to continue this path and to try to understand this humble RNA molecule. And thank you for listening. This is my wonderful group of students. This is our lab page, COBRA Lab, and you can find all our methods on GitHub. And these are our research focus areas. If you're interested, go at tiny.cc/COBRA or just search COBRA Lab, and you can find a lot of information in our lab. Thank you, and I will take questions.
I have a question. So you mentioned about the tertiary structure is not like we can shed light on tertiary structure by the secondary structure. So how easy to like translate these shapes into tertiary structure?
Mm-hmm.
Is it easy?
Define easy. But there are some existing programs that can take secondary structure and then give you the tertiary information.
Just like going from the primary to secondary is like a big deal because like you are literally checking every combination in between these and like tertiary.
That's not going to be that difficult.
So I don't think they would be able to hear you over Zoom. So if you can like just repeat this question.
Oh, okay. So the question was, is there any way to move from secondary structure to tertiary structure? And is it going to be as difficult as prediction of secondary structure from the sequence? So yes, there are existing programs that go from take the secondary structure and produce the tertiary structure. And no, it's not going to be as difficult as going from the primary sequence to the secondary structure. No worries. Go ahead.
So, like, pretty cool talk. So, I don't really consider that discovery quite hard. So, I'm wondering what's the limit to these predictions right now. So, I know there's some pretty complex structures RNA in the 5' PRs that are reinforcing many viruses. But I don't know, like, what's the length, especially that these models can handle right now. Or is that something that more like?
So the question is the length of how long of the RNAs can these models, computational models in general, handle, or just the methods that I mentioned in here?
The length and also the complexity. So you know the structure that have like multiple pseudonodes.
Okay. So the complexity. So the program that we used over here, iterative H-fold or Shapify, can predict a diverse set of pseudonautid structure. There was a study in 2006, I think. They looked at over 1,100 pseudonautid structures, and out of these pseudonautid structures, there were 9 structures that our method or this way of looking at the structures cannot predict. And out of these 9, 6 of them were not even predicted by the PNAS program of reverse genetics. So there are complex structures that we cannot predict. They're not that many. And in terms of the length of the sequences we can handle, because of n cube time and n square space, we can easily go to 1,500 bases, which is much higher than anything else that you can find with pseudonaught prediction. Normally, if you go with peanuts, for example, it's going to be in the order of 200. If you go with our naughty program, it's going to 400 bases, but even with the 400 bases, you need terabytes of memory. That's not going to be the case for iterative H-fold or for Shapeify.
Thank you for the talk. I was wondering how sensitive are structures to sequences? So like a small change to the sequence. Are there examples where structure has like changed immensely on just changing a few base pairs?
Yes, definitely. So there is, so the question is, if we have a small change of a sequence, can we have a global structural change? Yes. There are cases that a small change in the sequence, depends where it is, it can change the structure, of course, significantly. Of course, it's going to be more significant if your length is smaller. You can see that. But if you have like a very large sequence, maybe it's not that. It's not going to be global a structural change, right? It can be local to the area. But yes, the change has been shown to be functionally important as well. So the change of the structure can be significant and it can be functional.
So thanks for the great talk. So there's a big literature regarding the RNA secondary structure, and mostly it's our combinatoric optimization based on utilizing the minimum energy flow. There are recent works coming with using deep learning, trying to predict the RNA secondary structures. So my question to you is, how aligned these programs, let's say the models we had originally, which have been huge literature, and the new models that are coming from the deep learning, and is there a huge difference? And what are the drawbacks?
Very good question. That's one of the hot topics right now, and everybody asks about that. There are many methods coming up that do deep learning for RNA secondary structure prediction. Some of them have higher claims of their sensitivity and positive predictive value. There was a study, a third-party study for how well they perform compared to a traditional minimum free energy prediction. I think the study was published in 2021, and that showed the growth of base pair in deep learning method, at least the ones that they have looked at, was not linear with sequence length, which you expect it to be. It was polynomial with sequence length, which is not possible. The other drawback for deep learning method or any type of machine learning methods is that they are getting their information, they're trained on known information. If you have an RNA that is not well represented in your training set, or a feature that is not well represented in your training set, you will miss it.
Can I ask a follow-up question?
Yeah, go ahead.
So with respect to this, I have seen some iterative models over the time that basically when the RNA length increases, it chops it down and tries to use almost a iterative approach. You can imagine if I have a very long one, I can chop the RNA model, right? So it will still see or rebuild some sort of secondary structure with known features, let's say, like a energy model or whatever thermodynamics that you're putting in the model. So it's not an optimal solution when you're just chopping this thing, and we have used it even in the combinatorics algorithms we use. So what do you think that is going to be the way? Because we are going to face this problem of N6, right? Whatever the complexity that we're going to talk about, like the length increases, it's going to be very hard to predict.
Yep, I agree. So the method that you're talking about, we refer to as windowing or tiling. So we cut the sequence and we try to predict the structure for that sequence with overlap, and then we try to pull all of them together and find the structure that goes over it. Well, it works for a lot of different cases. I think RNA-Z actually has this approach as well. What it misses is the long-range interactions. So when you have long-range interaction, detecting the long-range interaction and resolving the long-range interactions correctly is going to still remain the same. It is an active area of research, and it hasn't been handled completely, but people are working on it. But yeah, I agree.
It's some post-processing after each iteration. I think that it has potential.
I think
That's local search, but
the challenge is it's not the optimal or suboptimal solution. So it's an 3D or some sort of an ad hoc solution that is on the
Yep.
I have a question.
Go ahead.
So it's a bit of like stepping back. So the RNA itself does this computation. Like we know that it, like in real life, in reality, it does this computation without using a supercomputer.
Yes, of course.
So is there like that makes me ask like a question, like maybe does it make sense that maybe the approach to solve some of these is not to try to solve it combinatorically, but to like try to simulate or emulate how this molecule behaves, and then go from there with some like stochastic like approaches. Is that an approach that works?
So that brings us to interactive molecules, right? In the cell, or a cell-like environment, there are many other molecules around this RNA, right?
Yes.
So one of the main limitations that I said about this model that we are using is that it assumes RNA folds in isolation or minimum interaction with the molecules, whereas in reality, it's not going to be the case. There are chaperones that bring the molecule, and there are some areas that are not accessible at all to fold, and there are other interactions in the molecule, right? So if that's why we are trying to incorporate known information. So if we have known information about blocking of a certain area of the sequence, or other types of information that we can get, like formation of a specific base pair over on the first part of the sequence, second part of the sequence, then yes, of course, the search space gets reduced, and then you can do that faster. This was the so in the hierarchical folding hypothesis, for example, it says that the sequence, the unfolded RNA, doesn't go back to a folded state in one go. First, the bases that are closer to each other fold back to each other, form the stems, original stems, and then when there are other bases are closer to one another, other base pairs that might include pseudoknotted base pair can form, or there would be some transition between the bases. Similar thing can happen when RNA interacts with other molecules. And as we have shown, we have reduced the time complexity for pseudoknot prediction from N6 to N-cube, right? If we include these kind of restrictions, of course, the asymptotic restriction is not going to change, but the search space changes, and the practical time for getting the structure out is going to be much faster than the one that we have over here.
So I was thinking more on like the line of, so I think that is very helpful, because that would speed up your combinatorial method. But then I was thinking more of like a physics energy engine, like where like you're thinking about all of these different molecules and thinking about like what do we know about the physics and chemistry of them pulling together, and like can we emulate that to some extent, maybe with some abstractions?
There are things, there are methods and research paradigms that look at that. It's called molecular dynamics. So they put a molecule within a solution, whatever that solution is, and then they look at the physics-based interaction between this molecule and environment. When there is a lot of molecules, the molecule that they can handle are within a smaller molecules, and then with a lot of interactions from the environment, they can predict the structure or interaction of this structure within picoseconds with clusters. So not practical at this time for longer sequences, but if you have a coarse grain prediction like the one that we have and try to go through molecular dynamics afterward, you can get a much better structure predicted.
Cool.
So you said this model works on like this practical for like 1,500 base pairs. So when you're talking about long range interactions in biology, like how long do you actually say that there's going to be effects? Like what distance actually matters to resolve?
I can't answer that exactly, but always remember that this molecule is not going to stay in a cell as a long sequence of string, for example, right? It has to become compact to stay within the cell. And then maybe some part of it gets opened up and some function gets performed, right? So compactness of this molecule is very, very important. And pseudoknots, for example, are the ones that make this compact interaction. So when you have some a structure formed already, the other bases are going to come closer to one another. So yes, they are. If the RNA was in a single line, they were far apart. But now that these structures are coming together to form some shape, these two ends maybe are closer to one another than you would expect, and then these interactions can form. If you ask the question the other way, how accurate is the energy model for these long interactions, then I can tell you that if you have, it has been shown by multiple papers that the structures predicted by many of the models that use this thermodynamic energy parameters are losing their accuracy after 150 base pairs. So yes, if the models become more accurate, we can predict larger ones, but you can trust the structure predicted for 150 bases, either for pseudoknot-free or pseudoknoted structures.
Okay, any more questions over Zoom? So I think we can take our final question.
Is there any promise for drugs that specifically target the sequence structure so that they don't
A very good question. That was actually the reason that we started for looking at frame shifting, because there was, for SARS, there was a study that showed if this pseudoknoted structure for SARS, for frame shifting in SARS, be targeted with small molecules, then it reduces the translation or reduces the translation efficiency for SARS, and that can be used as a treatment. The same thing was pursued for SARS-CoV-2, and that's why we have this research to find the functional sites so that if they are blocked, destroyed, maybe we can reduce translation.
Well, thank you so much.
Thanks
for the great talk, and thanks everybody for your questions. So we have some pizzas out here and some drinks. So please enjoy, and see you for next time around. Have a good one.
Nice talk.
Thank you.